My name is Edward Lau, and I'm here with my colleague Brian Bleakley from the HeartBD2K Center of Excellence at UCLA. In this webinar, we'll give a tutorial and demo on how to locate, access, and analyze a protein turnover dataset that was acquired at our center and recently made available to the public. I'll start by introducing various platforms we use for disseminating proteomics datasets. Afterwards, Brian will give a live demo on how to access the dataset in practice. There is now an increasing number of datasets available on public proteomics data repositories, with over 100 datasets being made available per month on Proteome Exchange alone. This is great for data reuse and also for supporting method development using the latest datasets available. But data sharing is also a central goal for our BD2K investigators and collaborators. We're hoping to help make proteomics data more accessible and valuable, and to avoid the situations where researchers may spend tremendous amounts of time and resources to acquire a dataset only to use it once and discard it afterward, which could lead to a loss of hidden information that may be extracted when different analysis routes or tools are employed in the future. Secondly, we also think of data sharing as consistent with good science and essential to allow research to be more transparent and reproducible. And lastly, we believe sharing data will help improve data visibility and promote cross-disciplinary collaborations. As the topic of this webinar is accessing protein turnover data, we'll introduce some basic information about the protein turnover dataset itself. The data contains a collection of mass spectra, protein identification results, protein turnover rate data, expression level data, and processing scripts. The protein data were acquired in the hearts of mice under normal conditions, as well as cardiac hypertrophy disease conditions. We have performed experiments in 6 mouse strains that are commonly utilized in biomedical research to help ensure that the protein data are generalizable. In total, the dataset contains the half-life information of more than 3,000 proteins and the relative expression of about 8,000 proteins based on label-free mass spectrometry quantification. The data covers 3 distinct subcellular fractions and over 200 non-redundant pathways, making it one of the larger protein dynamics datasets that are currently available. The challenge in sharing a big proteomics dataset is that a proteomics project may generate many different types of files, including raw mass spectra, identification results, quantification results, and other processed files. The spectral files are rather complex and not just linear sequence information, so the many types of files may not necessarily fit in a single repository, which might be optimized only for holding a certain type of files and data. So we have to devise a clear system as to where and which platforms the different types of files should go. First, there are the raw mass spectral data. These are the big spectral data files, and we have uploaded them to a stable and high-capacity repository. PRIDE provides an ideal place to access these files, which could also be discovered via the Proteome Exchange Consortium. They can be accessed to the location here. Secondly, there are the processed data files. These are not as voluminous, but they vary in file format and data structure. They are also the files that would be more readily readable and reusable for most data users. For these files, a proteomics data repository may not be appropriate, and we need a more versatile and dynamic solution. We have stored these files in Synapse, which is an open data analysis platform from our center collaborators at Sage Networks. Thirdly, we need a portal to tie together the different files and locations, and to show the users where to find individual elements and how to make sense of the data. For this goal, we have published an article recently on scientific data, an open access journal from the Nature Publishing Group that aims to promote open data and data science. In the next few slides, we'll briefly go over each of the 3 platforms and how they fit together as a general data dissemination mechanism, and how files may be retrieved from them. Accessing raw data on PRIDE. The raw data files can be accessed on a PRIDE archive project shown on the side here. This is a publicly accessible PRIDE project that contains approximately 1,500 raw mass spec files, and also the protein database search result files in sequenced format. project contains basic descriptions behind the sample processing and data processing protocols, lists the individuals to contact, and the metadata including cell types and instruments on the right. The dataset files themselves can be downloaded at the Download Project Files button on the upper right, which takes the visitor to a list of data files on the next slide. The listed files can then be downloaded either individually via HTTP or all at once via either FTP or Aspera Fast Download, with the links on the upper right. Alternatively, the raw files on PRIDE can be accessed via PRIDE Inspector, which is a standalone application that can be downloaded from EBI to browse PRIDE datasets. Users can then identify the dataset they wish to download by entering the project's accession ID, in our case PXD002870. It is also possible to manually download the entire project via Unix commands, as shown on the right on this slide. Accessing processed data on Synapse. The processed files are stored on Synapse, a platform built by Sage Networks. The investigators at Sage Networks have a tremendous amount of experience with open science, and so led in efforts including the Apple HealthKit research program, and more recently awarded a grant from the Precision Medicine Initiative. They have designed Synapse to be an online platform for processing data and integrating datasets and toolsets. Via Synapse, data users can download and upload files, read and contribute to wiki pages, track file provenance, and perform data analysis together. The Synapse interface is organized into the Wiki tab, the File tab, and the Tables tab. Here we're showing the view of the Files tab, and it can be seen that a number of subfolders containing different project files, including turnover rates data and tidy data tables, are listed here. Because the platform is file type agnostic, multiple types of data can be accessed at the same time. One advantage of this system is that users can access different types of processed data, including protein ID results and graphics, as well as data processing and statistical programming scripts. Users can also upload their own scripts back to the platform, so if somebody creates a plot that visualizes the data in a certain fashion, the script used to generate the graphics can be uploaded to the Synapse project, and other users interested in the analysis can download the code and build upon it in the future. Synapse also provides a wiki interface for users to access more detailed information on data acquisition and processing, as well as codebooks and data variables. The users can access different wiki pages displayed on the menu, shown on the upper left here, and users can contribute to the wiki record and add pages as they analyze the data. Well, this is an important component for us to share proteomics data on Synapse so that it serves as a platform not just for storing data, but also for consuming data. The wiki serves as an evolving document that can house crowdsourced annotations by the research community as future data analysis and reanalysis become available. For many use cases, raw data may not be the immediately most useful entry point. Raw mass spectrometer files are almost 1 terabyte in size, so there's the question of how often users can access them and rerun the entire analysis pipeline. Having only raw data will also limit the analysis opportunities to only users with a certain amount of computational skills and resources to reanalyze the raw data files from scratch. For most researchers, having a tidied data table may actually be more useful, which is what the users can find on Synapse. The data user could take the numerical values of turnover rates and plot them into their own models, or they could ask what the half-life is for a protein or pathway that they're interested in. So we believe there's a lot of value in sharing processed or semi-processed data. Another great feature of Synapse is the emphasis on data provenance, so users can see what scripts were used to generate data files, such as shown on the bottom here. You can see on the provenance box below the lengths of files and analysis together with the processes that are used to generate a particular file from other files. So that is consistent with the collaborative analysis and continuous analysis idea, where there's a data set available and different users can come together and validate and improve on the analysis if they wish to do so. Accessing documentations and instructions. Because the data exists in multiple repositories, it'd be great to have a central entry point to inform users on where all the data components reside. It will also be great if users can understand the motivations and scientific backgrounds behind the data, and have a way to cite the dataset in subsequent publications. This is where scientific data comes in. Scientific Data is an open access journal by the Nature Publishing Group that aims to promote open data science. We published a data descriptor article with the journal earlier this year, which can be accessed at the location shown at the bottom of this slide. The data descriptor article contains 5 major sections: background, experimental methods, data records, technical validation, and use cases. We'll give an overview of some of these sections in the next few slides. One of the first things the reader can find from the data descriptor is the background and motivation behind why the dataset was gathered in the first place. We won't go into details here, but protein turnover is now implicated in the development of many human diseases. But getting detailed information on how protein homeostasis can change inside the body can be quite challenging, because these processes don't necessarily alter protein expression. Whereas protein expression is quite straightforward to measure using mass spectrometry or even Western blots, with the temporal processes that we are interested in, it would require a way to distinguish old and new proteins in vivo and to measure the system at different time points, and to feed the data into a coherent model. So the investigators at our center have developed a technological platform. The schematic here shows how it is used to generate the dataset. The platform combines isotope labeling with heavy water in the mouse strains, shown on the left, followed by the extraction of proteins at different time points, followed by the measurements of isotope incorporation using high-resolution mass spectrometry, as shown on the right. Data were then analyzed by a computational workflow, as shown on this slide. The proteins represented in the mass spectra were first identified using the ProLucid database search pipeline. And then their abundance and half-life were quantified with the in-house software ProTurn, which uses a custom kinetic model to calculate the half-life of proteins inside living animals. With this technology, we have the opportunities to measure protein turnover rates and identify changes that accompany the development of heart diseases. The right-hand side of this figure shows the data dissemination workflow, with the data descriptor manuscript essentially serving as an entry point that directs users to different platforms. The data descriptor article also shows technical validation steps that were carried out for the dataset. Because most data users won't start from the rawest mass spec files, it would be great if they can be assured about the soundness of the initial data processing steps, and to see figures that demonstrate the QC process. For the data here, we considered how well the kinetic model fit to the data points. For example, the figure in panel B shows a range of fitting scenarios from R² of 0.99 to 0.8, the latter which we deem as the minimal acceptable value. We can see how much of a data variance remains after each step of filtering in each sample. We can also explore the relationship of what determines data quality. Here, panel C shows the relationship between the number of time points in which a protein was quantified to the proportion of peptide time series that conform well to the model, so users can make an informed decision on how they want to filter the data and so on. Although these QC figures may sometimes not receive the attention they deserve in a regular manuscript, or may be buried deep inside a supplementary information file, we think they are a great resource for data users because they show the distributions of data points and what happens if analysis parameters are altered. Finally, the data descriptor also contains example use cases and data analysis routes. In the first example use case, the user can look up the turnover values of a protein of interest. For example, a cardiovascular researcher may be interested to get information on, let's say, mitofusin to understand mitochondrial turnover and calibrate fluorescence timers. In the second example, the user may look up the turnover distributions of pathway or an organelle. For example, A basic scientist studying the Golgi can look up the regulation of proteins in the Golgi apparatus. The data can also be combined with other datasets to discover new disease signatures. Thirdly, we think an omics scientist can reuse the mass spec data to identify new protein sequences and understand their half-life. For example, if there's a new workflow to identify single amino acid polymorphisms, the turnover rates can be reanalyzed from the data. Fourthly, we think the data can help method development in protein turnover studies and guide future experiments. The data can be used to compare protein turnover across animals, labeling protocols, and methods. And data scientists can use them to develop new fitting methods or software. Lastly, to promote usage, we've also created step-by-step instructions on the COPA-KB website, which contains instructions to open up the data files in Excel or in R, sample codes, and screenshots of the intended results. With that, I'll hand over to Brian, who will now perform a live demo on downloading and querying some of the stored files following these instructions.
Hi, I'm Brian Bleakley, and I'm going to be covering the use cases that Edward mentioned. These use cases can be found on the COPA-KB website. Just click the Synapse icon in the top right corner to go to this page. The first use case I want to cover is retrieving the turnover rate of a specific protein from Synapse. So the first thing I'm going to do is navigate to the Synapse project page. The default takes me to the wiki page where I can see the overview here. I'm going to click on Files to go download files. In the Tidy folder, you'll find the most cleaned up versions of the data that are ready for use. And I'm going to download all protein K. Here I can get a preview of the file. I can download the entire thing to my desktop. Okay, once the file is downloaded, I can open it up in Excel or another spreadsheet program. And here you can see the data, the unit product session numbers, the mouse strain, the group, and the K values. If I go to the Data tab of Excel and select all the data and click on Filter, it'll automatically identify column headers, and I'll be able to sort and view based on unit product session number or based on mouse strain. Now I will demonstrate how to reanalyze mass spectrometry raw files using ProTurn. First, follow the link to the project archive in PRIDE. Next, click on Download Project Files. Now I can see a list of all of the files displayed here in the browser. I can click to download one. These are large files, and they'll take several minutes to download, so I will skip ahead to show you one that's already downloaded. As you can see, this is a raw file format, so it must be converted into NZML to be compatible with ProTurn. So I can use ProteoWizard to do that. I open it up in MS Convert GUI, select the output format MZML, and click to start conversion. It's now converting the file. This process will also take several minutes, so we'll skip ahead until it's complete. To analyze the data files with ProTurn, both the search output files, which are DTA files in this case, and the MZML files must be stored in a particular folder configuration so that ProTurn can identify which files are associated with which time points and which fractions. Inside the MZML folder, there must be one subfolder for each time point. And inside of each time point folder, there must be one folder for each MZML fraction. Within the DTA folder, there must be one DTA file for each time point. Now, we will perform the analysis using ProTurn. I'll launch the ProTurn GUI, select the file format, DTA in this case, select the data directory, set my integration parameters, set the number of threads, and start the integration. This is a sample data set that will integrate much faster than the full data set. So it'll be done here in just a few seconds. Once the integration has been performed, we can move on to fit kinetics curves. It still remembers the data directory I was using, and then I can set my fitting parameters and fit kinetics curves. When that is done, the data will appear here in files HLOut and HLData.out. These are tab-delimited data files that can be opened up in Excel. It'll automatically identify the tab-delimited format and present it here in columns. And those columns can be filtered and sorted in the same way we demonstrated with the Synapse files. That concludes the overview of our protein turnover data set on Synapse. If you have any questions, please contact Edward Lau or myself via email, and we will be happy to help you.